All right, so last time we talked about the singular value decomposition of the stoichiometric matrix, and we were introduced to the fact that orthonormal basis vectors may not be the best way to span the spaces associated with the stoichiometric matrix. So today we're going to talk about a different set of basis vectors that actually correspond to pathways. And these are the basis vectors we like to use to describe the null space of the stoichiometric matrix. So we're going to be focused now on just the null space. So first I want to talk about, in general terms, about what acceptable basis vectors for S look like, and acceptable from a biological standpoint. And I'll compute a few set basis vectors here for simple pathways from textbooks that we are all familiar with. That will then lead us into this topic of convex analysis and looking at convex basis vectors that are used to span finite spaces, because all real variables, physical variables, are finite in the numerical values they can take on. Therefore, we are always interested in subsets of these large linear spaces, such as the null space that is associated with the stoichiometric matrix. I'll talk just a little bit about extreme pathways, which are convex basis vectors. And it turns out that they are numerous. They are too many to be of interest. But there's a subset of them we can calculate, which is called the MinSpan. And this leads us to a biologically meaningful subset of extreme pathways that we can use to interpret biological data types. And these are my examples. And then at the end, I want to put the material in this text a little bit in context, both historical context and the fact that we now have mathematical objects or definitions in systems biology that correspond to classical definitions of concepts in biology. So we can now mathematically represent those fundamental concepts like pathways, and we can compute with them. Okay, so first let's talk about basis vectors for spaces, and the null space in particular. So that image that is shown in this slide, if you look down the left side of it, you see something very familiar. An XYZ coordinate system that may be used in fluid mechanics to describe a flow field or something like that. And the mathematics are normally described in terms of these basis vectors that are often called I, J, and K. And if you want to locate a spaces in a point in a three-dimensional space, we have to add these vectors up in a particular way to get that point. And those are the weights on these basis vectors, here indicated by A, B, and C. And below that, I show what might be called the loadings or the spectrum of loadings on those basis vectors to get that point. So A, B, and C take on particular values as you move along those dimensions in the space to locate that point exactly. Now what we're going to talk about is something similar to this, except we're going to be dealing with convex spaces, like shown in the top right. And we're going to be looking at pathways going through a network, like the image below shows. And there are going to be many of these. So it's going to be the dimensions of these spaces we are dealing with is much greater than 3. So when we look at the spectrum of weights, it can be almost continuous, like shown on the bottom. If you have 100 basis vectors, you need 100 loadings or something like that. That spectrum of loadings becomes much more complicated than for a three-dimensional space. So that's kind of to set the conceptual framework relative to something that we are all familiar with. All right, so let's talk about finding basis vectors for the null space of S. So here I have a very simple system that we're going to use as an example, where we have 4 nodes or 4 compounds in there, A, B, and C, and we have 6 fluxes associated with them. So there are 6 reactions in here, and this matrix is therefore 4 by 6. It's full rank, so it's the rank is 4, And since I have 6 columns and rank is 4, the dimension of the null space will be 6 minus 4 or 2. So we're going to be looking for 2 basis vectors here to span the null space. And the null space will represent all the possible flux states of the system. Now you can calculate a basis for the null space in many different ways. If you just put that matrix into Mathematica and ask for the null space, you get these 2 vectors. And they are represented now as column vectors, which is what they are. They are 6 by 1. And if I multiply them into the stoichiometric matrix from the right, I will get a 0. And we'll get to that in a moment. But here's a set of basis vectors that Mathematica may give us. First thing I want to impress upon you is that the stoichiometric matrix is a connectivity matrix. And we're calculating all the flux states of it. So these are all kind of, the basis vectors all become paths in the network. So we can actually visualize these mathematical basis vectors on a map. And the 2 basis vectors shown by these 2 column vectors on the right can be represented as paths through that network. And you see first that the first column has 1, 1, and 1 on the first fluxes and 0 and 0 on the 5th and 6th. So if I draw that out, it's a balanced set of fluxes into the network through the upper branch and out. That's what that basis vector is. The second basis vector, if you look through the weights on the fluxes in that second column, you see that that represents a circular path inside the network that we're looking at. So the basis vectors can be represented as pathways in the network of interest. Now, any steady state solution is in the null space. So S dot any flux vector in the null space would give me a 0. And these 2 basis vectors, R1 and R2, are in the null space. So if I multiply them with the stoichiometric matrix, I get a 0. And any linear combination of these basis vectors, R1 and R2, will give me a point in the space. So if I pick any weights, W1 and W2, and I add up my basis vectors, I will get a steady state flux vector that is in the null space. So we say that R1 and R2 span the null space because I can represent every point in that space with the appropriate weights. So let's just take an example of that. So here is one particular flux distribution where something is flowing in at the rate of 2, whatever our flux units are, and there's an even split of A down the pathways leading to B and D. They converge again and leave the system, and the flux out is 2. So in a steady state here, the flux vector will be 2, 1, 1, 2, 1, 1. So that's our vector. Our steady state flux vector, sometimes we call this the flux distribution because this is a distribution of weights in the flux vector. So we can take that vector 2, 1, 1, 2, 1, 1, and we can break it down into those 2 basis vectors that I had before, and that's what's shown in this slide. And this particular flux distribution is calculated by multiplying the first basis vector by 2 and the other one by 1, summing them up to get that point. What I also want to impress upon you is that we can almost do that addition visually. So on top there I show the 2 basis vectors, 2 times R1 going through the system and 1 times R2. If I just superimpose them on one another, we can see immediately what the addition is to give us that flux distribution. Now, it turns out that we are very happy with that first vector. It looks like a pathway coming in and going out. But we are not happy with the second vector. It is a legitimate vector mathematically representing that loop. But we don't like that vector because it has these weights of -1 in it on fluxes 2 and 3 and goes backwards through a set of irreversible reactions. So it's describing a flux that goes against thermodynamics. So we don't like that basis vector, but we like the first one. Now, it turns out that there's an infinite number of set of basis vectors to choose from in principle. So we can now just get rid of that second basis vector and pick a new one. So you can do that by a very simple combination of them. And if I take these two basis vectors, post multiply by the 2 by 2 matrix, I can get a new set of basis vectors. So the column, the first column in that 2 by 2 matrix will tell me how I combine R1 and R2 to give me the new set that I'm going to call P1 and P2. Since I like R1, I'm just going to put 1 there and 0 on the second vector. So I put 1 and 0 in the first column of that 2 by 2 matrix. B1 just becomes equal to R1. I don't do anything to that. But to get rid of these two minuses, I could simply add up R1 and R2. And then they will go away. So in the second column in that 2 by 2 matrix, I put 1 and 1. And so I just add those two up to get my second basis vector in the new set. And now I have a vector that is 1, 0, 0, and what is that? 0, 0, 0 in the second column. Now I have a set of new basis vectors. I've just done a transformation on my basis. And I picked one of the vectors from my first basis, and I formed a new second basis vector in here. So what is nice about this second set of basis vectors is that I can add them up in a positive combination, and I can always get positive fluxes through all the irreversible reactions in there. So I don't have to violate any thermodynamics using this basis vector, and I can add them up in a non-negative fashion. So these two weights now are greater or equal to 0, but they're never negative. So I never run anything in the backwards direction. And if I plot them out now, they look like two pathways through the system. And it's quite intuitive and immediately obvious that any linear combination of these two will give me that split properly and that reconvergence of the pathways before they leave. So this shows us a few things. First, the basis vectors in the null space are actually pathways. Since there's an infinite number of choices of basis vectors, in principle, I can pick an infinite number of pathway vectors to look at. But this example shows that not just any choice of these basis vectors is good. So we are now going to talk about what is a good set of basis vectors in the null space that is consistent with biochemistry and our understanding of network function. Before I do that, I want to just go through a few examples of how these how we can compute these basis vectors and how we interpret them. So one example that we showed in the chapter on the stoichiometric matrix is glycolysis, and glycolysis as a system. So there are inputs and outputs, and I can balance all of these by putting S dot V equal to 0. And I believe we have this slide there, this table there as well. This is the annotated stoichiometric matrix where I put a few things in there, I guess the connectivities and the participation numbers. I did the elemental balancing as we talked about, but now I have an additional set of rows in there designated as P1, 2, and 3. It turns out that the dimension of the null space of this matrix is 3. So there are 3 dimensions in here I need to span. And I've chosen these basis vectors that are shown there, and I will show them graphically here in a moment. I chose them, but they're also pretty close to what some algorithms will actually calculate for you. Any linear combination of them will give me a flux map. And there's a particular flux map given there at the bottom that is consistent with an experimentally determined state of glycolysis in the red blood cell. So this actually represents a realistic system. Now, here we are showing what these pathways represent biochemically. I chose them, remember that, myself, based on intuition, but we're going to also see that these pathways are consistent with some mathematical definitions. So the first pathway in there represents the flow of glucose in and 2 lactates going out, and there are 2 ATPs being made that can be then used to drive a process somewhere else in the network. So the dash boundary is a virtual boundary. The solid lines should be actual boundaries. So that line there on the right should probably be a solid boundary. The second pathway is the lactate-pyruvate exchange. This is lactate going in, making NADH and pyruvate coming out, and then that NADH can be used to satisfy a redox load. And the final pathway here is a little artificial in this system. It's just ATP going in and coming out. And we'll look more at this later on. So here's that block I added at the bottom of the stoichiometric matrix in the previous slide, highlighting these pathways. And if you look at the second pathway in here and its weights, it'll show the uptake of lactate, the secretion of pyruvate, the generation of NADH, and then the utilization of that redox potential. So this is a functional module in this pathway. And I think you can immediately see, if I'm observing an experimental system where, say, 2 glucose come in, and 1 lactate comes out, and 1 pyruvate comes out, that flux state would correspond to the summation of these 2. So these are 2 different modular functions of the system. One is the generation of high-energy phosphate bonds. The other one is generation of redox potential, and I can combine them in any ratio that I like. Oops, missing something. It generates a proton also, this pathway. Now, I can look at a slightly more complicated system. Here is that same glycolytic system I had before, but now I have fused with it the pentose pathway. And that's the pathway that's in this box. So now glucose 6-phosphate will flow out of glycolysis into this pentose pathway. Fructose 6-phosphate will come back, and GAP will come back. But those are common nodes between these 2 pathways, so we can just fuse this into one system. I think we talked about that process before. Now my matrix looks a little more complicated, a bunch of numbers in here, but I'm going to show the same thing as I did before. I've appended to this matrix 4 pathway vectors, because it turns out that the dimension of the null space is now 4. So when I add the pentose pathway into the glycolytic system, I get a stoichiometric matrix that has a null space of dimension 4. I've also chosen these pathways, and they are shown in this diagram over here. And the first 3 are identical and the same as I had before. There is one new dimension that comes into the null space here that goes through the pentose pathway. The reason I picked this example here is that it shows that the pentose pathway itself isn't a separate pathway. When I look at it as a system, it's a separate chapter maybe in your biochemistry textbook, but once it's integrated with glycolysis, it gains an integrated functionality that is a combination of the state of glycolysis and the pentose pathway. And this particular pathway will consume a glucose. A portion of it goes through the pentose pathway to produce CO2, and eventually all the carbon comes out as lactate in the end. So this example is picked here to drive the point home that pathways in the null space have to balance the whole network. You cannot just look at the piece of the network and think about that as being separate from the rest, because it isn't functionally separate. Now I can further expand this example by putting in the synthesis of A and P. And now what I have to call out are the purine biosynthetic pathways and the purine degradation pathways, and they are shown in there. And these happen to be the pathways that are in the red blood cell. So this is now a whole network that has a bunch of deaminases and dephosphatases. It has a salvage pathway and a few other things in it. And I can build a stoichiometric description of this, and I can seamlessly fuse it into the network that I just looked at that was comprised of glycolysis and the pentose pathway. So it's the same process, just gets a little more complicated. There's the stoichiometric matrix, and I annotate a block to the stoichiometric matrix of pathway vectors as shown at the bottom. In this case, there are 8 pathways. By adding the purine biosynthetic pathways and degradation pathways, I add a dimension of 3. to the null space. It was 5 before, now it becomes 8. And I've chosen pathways here as these basis vectors as well. So what happens here is that I still have my same glycolytic basis vector as I had before. It's preserved between these null spaces. I have the pyruvate lactate exchange. That's also the same. I had that integrated pathway with the pentose phosphate pathway I had before. But that simple pathways of A and P coming in and going out breaks down into 5 dimensions in the space now. And 3 of them are A and P degradation pathways. And one is called a salvage pathway, which recycles the pentose to rebuild the nucleotide. And then there's a futile cycle in there. And that's a pathway that is often described in biochemistry textbook. That is a pathway that actually degrades a resource like ATP. So that's what happens over here. And the point of this example is to show you that there are 5 dimensionalities in this 8-dimensional, there are, 3 dimensions in this 8-dimensional space that kind of represent the same thing, A and P degradation. So they are mathematically distinct, but biochemically and functionally they're related. And actually they will be oblique, as it turns out, and have similar dimensionality in the stoichiometric, in the null space, and can kind of be bundled together to represent a biochemical functionality that you then can fine grain if you want into each one of those dimensions. Okay, so that gives you an example of what these pathways might look like. They're relatively easy to interpret for simple systems. This last one became a little too complicated perhaps, and you can kind of pick them if you like intuitively to make biochemical sense. But that becomes increasingly difficult with scale. Even when you get to the core E. coli network that we use in this class, it gets too complicated. It's hard to go through the 23 dimensions of that null space and pick good, meaningful vectors that represent biochemical function. So we would like to do this algorithmically. We'd like to be able to state mathematical criteria so we can now calculate pathways. All right, so to do that, I need to introduce you to convex analysis, convex spaces, and convex basis vectors. So first, let's remind ourselves as to what is a convex space. So a convex space is defined mathematically in a very simple fashion. Pick any 2 points in that space, and if you draw a straight line between them, that entire line will be in the space. What it basically means, if you're standing in the space, you can see it all. There's no hidden part of it. And in fact, we'll see later when we do optimization in a convex space, you're guaranteed to find the optimum because you can see everything from any starting point you pick in the space. Now here's another space that is not convex. It has this indentation in it. So if I stand at point A, I can't see point B because I have to look outside the space. So if I draw that straight line, a segment of that line will be outside the space. So this is not a convex space. And this is an example of a situation where you're trying to find an optimal point in a space. You may not be able to find it because you can't see everything from your starting point. Okay, so that's an illustration of what a convex space is. It basically becomes a polytope in high dimensions. And what a non-convex space is. And a non-convex space is normally bad news. But fortunately, we're going to be able to deal with convex spaces. So let's talk a little bit about forming a basis for a convex space versus a linear space. And the convex space gets formed for us out of the null space of S because the fluxes cannot take on any dimension, any numerical value, I should say. They are non-negative and in fact at a finite maximum value. So that leads to the definition of the convex space. So here's a table from the book that represents the 2 spaces and the contrast between them. And I just want to pull out 4 items from this table that are important. First is the definition of a space. A linear space is defined by a linear set of equations. S dot V equals 0. That would define the null space for me. But the convex space adds inequalities. And that's what's shown in that equation, namely that each one of the flux values will have a minimum value and a maximum value. Can't go past those limits. So that's what closes up the space. Now we're all familiar with a set of basis vectors for a linear space. You pick any three linearly independent vectors in the space, and any linear combination of them will land you somewhere else in the space. And that includes negative weights on the basis vector, which we saw earlier we will not like, say in the case of an irreversible reaction. Now, when we span a convex space, we calculate a set of basis vectors called P, and the weights we put on those basis vectors cannot be just anything. They will have a restricted range of numerical values that are consistent with the bounds that we place on the space. So that's our second property. The third property highlighted here is also important. For a linear space, the number of basis vectors we pick is equal to the dimensions of the space. In a three-dimensional space, we need 3 basis vectors. In a convex space, to span it, we may need more vectors than the dimensions of the space. And we'll see this in a moment. And the fourth property that is really, really important says that the basis for a linear space is not unique. You can rotate that basis and you still have a basis for the space. But the basis for the convex space is unique. It may be of higher dimension and there may be more vectors in it than the dimension of the space, but it's unique. That basis becomes a straight property of the equations. And in this case, it's the flux balances defined by the stoichiometric matrix, then bounds on the fluxes, which are put down by thermodynamics or enzyme capacity constraints. All data. So that set of equations represents data. It defines a convex space as a unique set of basis vectors. And we're going to see those basis vectors are pathways. They cannot be arbitrarily chosen. And this is extraordinarily important. That the basis vectors become network properties, not something you dream up. Okay, so let me show you now a little bit about the second and third property. Not going to prove those mathematically. That's the subject of a class on convex analysis, but just illustrate them to you. So here's a pyramid, a 4-sided pyramid. This is a 3-dimensional object. Now, it turns out that if I want to represent that point A at the bottom of the pyramid, it depends on where that point lies, I may need 4 basis vectors to get to that point. Because otherwise I may have to drive one of these basis vectors in the negative direction and then back into the space to get to that point. But that's a no-no for me. So if I want a convex basis and a non-negative combinations of that and land inside that space, I will actually need 4 basis vectors here. So to represent all points A in that space, I need 4 basis vectors. That actually turns out to be these edge vectors, as I'll show you in a moment. And that P should probably be changed to a lowercase because it's a vector, not a matrix. And what I'm showing at the bottom is that the selection of the weightings designated by these alphas is not arbitrary. They fall into a certain range. Okay, so property number 4. Here I'm showing a triangular segment or a cone in a 2-dimensional plane that lies in the first quadrant, that white area. The blue areas are outside. And I'm also showing a point A that is inside that space. And I'm showing you 2 sets of basis vectors here. One I'm going to call P1 and P2, and they lie on these edges that are shown there. If I add these vectors together with positive weights, I think you can immediately see that any combinations of those two gets projected from the edges into the space. If I put a negative weighting on those vectors, I will leave the space. Okay. Here is a set of alternate basis vectors that are called B1 and B2. One of them lies inside that space, one of them lies outside that space in the blue area. Now, if I want to represent that point A, I'm going to have to drive B1 in a negative direction and then add B2 in a positive direction to get to the point A. So I have to put a negative weight on one of the basis vectors, as shown here. Now, it turns out that P1 and P2 are unique set of basis vectors that span that convex space, and they lie on the edges. And that's where that uniqueness property comes from. Not proven here by any means, but just illustrated. Okay, so let's keep that in mind. We think about now convex basis vectors at the edges of a convex space, some polytope in a high dimension. Now let's look at a little bit about how these finite or confined null spaces are formed. They're very hard to draw in 3 dimensions. You cannot do too many of them in 3 dimensions. And really the only thing you can do is for a simple flux split. So you see a compound A there that is produced by one reaction and degraded by 2. And each one of these fluxes will have a finite range as indicated. They have to be positive, and then there's a Vmax on them. And those are set at numbers here 6, 8, and 10 for illustrative purposes. Now, since these fluxes are either 0 or Vmax, can only take on those values, they define a box that is shown here. So any point in that box will be a flux state that will satisfy those constraints. I have to be in that box. And many of them are not steady states. But if I add the steady state equation here, that is shown on top there, which ties the 3 fluxes together by an equation, I define a plane. And that plane is shown there in red. And that plane goes through the origin. And this is a plane that the row vector there is orthogonal to. Remember, all the steady states lie in a plane that's orthogonal to the row vectors in the stoichiometric matrix. This row vector here is just -1, -1, and a 1. So the intersection of that plane through that box represents my confined null space. And that's how these null spaces are formed, these confined null spaces. There's a hyperbox of some sort based on constraints on the individual variables. Then there's a hyperplane that goes through that box. And that intersection represents my confined null space. So let's look at this a little more accurately. So there is my flux split with the constraints. And then I have a 3-dimensional image of what I was showing before, now actually scaled. So I have that box, and I have that plane going through it, and that dark area represents the portion of the plane that's inside the box. And the pink-colored box there to the top right shows the linear combination of P1 and P2, my basis vectors, and the alphas. And the alphas are restricted as shown there. Alpha 1 between 0 and 6, alpha 2 between 0 and 8, and the summation of alpha 1 and alpha 2 has to be less than 10 to observe the restriction on V3 being less than 10. Now, I can take that intersection is just a plane, and that's what's shown in panel C, and the intersection is actually that green segment there. And it's formed by just observing so there's the plane, and there's the box. And here are my 2 basis vectors, which are actually just pathways through there. Basis vector number 1 is just 1 on V3 and 1 on V1 going out. And P2 is 1 on V3 and 1 on, must be V2 going out. So those are my 2 basis vectors. They're just the 2 pathways that can go through that flux split. And any combination of them are in that plane. In that triangular segment that will be defined by this green swath there, but it's going to be constrained. So here I'm showing the constraint on V1, here it is on V2, and here it is on V3. And when I observe all these constraints on the flux values, I form that green box. So that's how we form confined null spaces. Okay, so we're going to be interested in those confined null spaces and how to span them with these basis vectors and how we actually do such spans. I want to introduce you now to the extremely important concept of redundant and dominant constraints. So here I'm showing exactly the same system. I have my two basis vectors coming in into that split, and I have the constraints on them. So I'm designating one in, or indicating one in green and one in blue. So hopefully this color coding comes out right. So if I am going only through the blue vector, B1, I can go 8 units along that axis before I hit a constraint. I can't go outside that. If I go on the green vector only, the B2, I can only go 6 units along that before I hit a constraint. Now if I add them two together, I hit that vertex at the top of this, I guess it's a trapezoid segment, which says 8 times B1 plus B6. But that would give me 14 units on the input, and V3 can only take a value to 10, so that's why I have that dashed line cutting through the space. That little triangle in white, with the green point as the apex is excluded because of the constraint on Vmax. Now let's suppose that Vmax, that enzyme is expressed higher, so now that Vmax is 16. Now that constraint that corresponds to V3 is outside the space. That means it's a redundant constraint because the constraints on V1 and V2 already define the space for me. If it's 10, it's not a redundant constraint. Now if it drops to 5 and goes through that last dashed line, it becomes the dominant constraint, and the constraints on V1 and V2 are redundant because they are subordinate to that constraint of 5. So this kind of graphically illustrates the important concept of redundant and dominant constraints. And this in turn means that we often need just few and a finite number of constraints on a genome scale model to close off the space. And essentially always, if you have constraints on the inputs and the outputs, which are experimentally measurable, you will close that space. Okay, so what does this mean? So there are all sorts of interesting attributes associated with these redundant and dominant constraints, and those are illustrated here. So I show in panel A these 3 pathways going through that little network there, and the confined null space, that cone, it actually turns out to be a cone in high-dimensional space, here in this case 3-dimensional space. And if I had the same Vmax on each one of these axes, I close off a 3-sided pyramid, basically. Now, panel B shows a different situation. And let's say that we were analyzing inborn errors in metabolism. And one of the reactions there, V3, let's say that represents a gene that is defective. And the Vmax through that reaction is much lower in a person with that inborn error than what we would consider a healthy person. And now you can see that the space is much smaller. It's much smaller because that V3, when you take it just as a slider and draw it down, you exclude a huge chunk of the space. That chunk of the space will be excluded as a possible flux state for a person with that genetic defect. So this is an illustration of how meaningful biological analysis can be carried out simply by looking at these constraints. Okay, so that's the notion of dominant and redundant constraints. So, a redundant constraint for most of us may become dominant for someone with a particular genetic makeup. And that may exclude what we consider a normal physiological state, at least to a pathological state. Very important concepts. So let me now talk about the extreme pathways. So I have said that the null space vectors, basis vectors, are pathways. They correspond to pathways through a network. I've told you once I form the confined space that those pathways, that the basis vectors are the edges of that space. And because they are the extremities of the space, we call them extreme pathways. They really represent extreme states that network can take on. So that's where that name extreme pathways comes from. So I just want to emphasize again something I said before. This is kind of pictorially how they are derived. You have a network, you reconstruct, get the stoichiometric matrix, you impose the constraints, and you get that space. And that space is defined based on the data, the information, the knowledge that goes into the reconstruction. So these pathways are not arbitrary. They are properties of what you put together. And they're called extreme pathways because they're at the extremities of the space. You can't go any further. Okay, now a lot of work has been done on extreme pathways and these sorts of pathway vectors. And I'm just going to talk now about a few of their attributes. You can classify them in different ways. And you see that these three icons below there, they show three types of extreme pathways. The first category is called a through pathway because it has an input and an output, and the result of that pathway generates some resource internal to the cell that can then be coupled to another process in the cell and drive it. So it produces what we call currency metabolites, these cofactors that are now charged with some property, high energy phosphate bond, redox potential, ammonium group, methyl group, or whatever. They can then be deployed to drive something else inside the cell. Type 2 is what is called a futile cycle, because this is an internal loop that will dissipate one of those currency metabolite pools. And these do exist, and they generate heat, for instance, so they can be used to generate heat. They are irreversible, they're only unidirectional. Because if they were to go the other way, they would be thermodynamically infeasible, and you can charge a pool for nothing. So they're unidirectional. And finally, there's a type 3 pathway in there that is a purely internal loop. And the flux through these has to be zero, basically by Kirchhoff's laws, right? Second law. You can't run a loop internal to a system because it'll dissipate some energy, and that can't happen. Okay, so you organize the pathways into a matrix where the columns are the pathways, and the rows in it will correspond to the fluxes. And you partition these fluxes so that, let me show it down here, you partition your stoichiometric matrix when you calculate these things, that the first block in it, the first set of columns in the stoichiometric matrix are only internal reactions. The second block will be the exchange reactions with the environment. And the third block will be these internal exchange reactions across these virtual boundaries that we draw as concepts, like I had for glycolysis before. And if I take this matrix P and I multiply the matrix S from the right, and that's a dimensionally consistent multiplication, I get zero. So the number, the rows in P are the fluxes, the columns in V are the fluxes, and they match exactly. So every single one of these basis vectors forms a perfectly balanced map, and it lies at the extremities of the solution space. Okay, so type 1 are of greatest interest. Those are the through pathways. Type 2s are futile cycles. They are unidirectional, and most likely all the genes in them are not going to be expressed under a given condition, so they're not functional. There are some hypotheses out there that say that they are intentionally regulated in many conditions to generate heat, to regulate temperature inside an organism. And type 3 are thermodynamically infeasible by Kirchhoff's second law, and they can carry no flux, and they can be eliminated. That part of the solution space can be eliminated. Solution space, now I'm talking about the confined null space. They can be eliminated by a variety of methods, and one of them in the Cobra toolbox is called loopless FBA. It eliminates those loops in your solution. Also, if you run the most parsimonious FBA solver, it'll eliminate all those loops too. So there are ways to get rid of them, but they can be there. And that little map there shows the first discovery of a futile cycle in glycolysis, namely that the phosphofructokinase will take glucose phosphate, 6-phosphate, and make 1,6-bisphosphate by deploying an ATP. But gluconeogenesis that goes in the opposite direction uses a phosphatase that just cleaves that bond off. So if both these enzymes are active at the same time, they're just dissipating ATP by going back and forth, back and forth, back and forth. And this was a puzzle early on in the discovery of metabolic pathways called the futile cycle. And like I said, you would expect under most cases that they are not expressed by, either one of these genes is expressed but not both, or they could be used to generate heat for thermoregulatory purposes. So that's the notion of extreme pathways. It turns out that when you go to a higher and higher scale with stoichiometric matrices, the number of these convex basis vectors blows up to an incredibly high number. And it's an NP-hard problem computationally to get them all. And that's because when these solution spaces get big and multidimensional, there are all these little nooks here and there. To go into them and span every little part of them, you need a lot of basis vectors. So that became impractical to have those. So there has been some work done in recent years to get a meaningful set of basis vectors, such basis vectors, that is practical. And one of them is called MinSpan, which I'm going to talk about here, done by a student here at UCSD. There was a group in Australia led by Lars Nielsen that had a similar definition called SLIPs, which were presented at a number of conferences, but I believe have not been published yet. So here is the idea behind the MinSpan. And this may get some of you more excited that like algorithms. So the issue is to find that matrix N that is shown in panel A, and any matrix that multiplies S from the left that gives you all zeros. So any matrix that fits that criteria will be a set of candidate pathway vectors. But we don't want just any one of them, like we've talked about. Now we're going to add an additional criterion here. We're going to try to sparsify that matrix as much as possible. What does that mean? That means that matrix N that I calculate to become my P should have as few non-zero elements in it as possible, because that will lead to the simplest set of basis vectors. And the algorithm that does this is a greedy algorithm. It turns out this is unique only in the following sense. You calculate the first basis vectors, and you make it a basis vector that has the fewest elements in it. And let's say it's 3. Rarely is that just one choice. So you can maybe get 10 basis vectors that have 3 elements in it. You pick one. And then you go to the next vector, and you sparsify again. And let's say that the next vector is a dimension 4, or has 4, I should say, non-zero elements in it. And then be another choice of 10 there, but you pick one. So you have to pick them as you go through them. I believe if you go through all these possible permutations of that, you start generating maybe not the full set of the extreme pathways, but you may catch most of them. I believe that Arash says that even when you place the less basis vectors in there, and let's say it has 15 non-zero elements in it, there are still extreme basis vectors that may have more than 15 non-zero elements. So this may not capture all the possible extreme vectors, but most of them. But as you lay them down with this algorithm, you pick a particular set. And the consequences of those choices, as you formulate the columns, has been studied in that paper. And it is it turns out that the choices you make are not that consequential to the ultimate conclusions that you reach. So the system there at the bottom has 4 MinSpans, and they're shown as individual basis vectors in panel D. But the good thing about the MinSpan is it calculates n minus k basis vectors, exactly the dimension of the null space. I think if you wanted to, you could probably get more, but the MinSpan is the minimum number of vectors that satisfy the criteria that I have stated. Now, you can now probe the biological meaning of these basis vectors beyond, saying that they're satisfying pathways on a map. You can start to look at the genes and the macromolecules associated with every reaction that you place in a pathway, extreme pathway. And panel A shows what we are all familiar with. is GPR. So associated with every reaction in a pathway is a GPR, genes, proteins, and so forth. And if you take a pathway, you have a string of these reactions, and now you can define a set of protein peptides, you can define a set of transcripts, and you can define a set of genes that have to be expressed. Now, remember, we talked about the fact earlier that these GPRs represent a container into which you can map omics data. And now I've used these pathways to define a set of GPRs that need to be coherently functional, or represent coherently functional proteome and transcriptional regulatory network to actually correspond to a function in the network. And you can calculate these as shown in panel C, and look at the properties and compare them to available data sets. For instance, you can map protein-protein interaction data sets on there and see if all the protein that correspond to a pathway are known to physically aggregate. Because pathways are known to be sometimes represented by a set of protein that are aggregated at one point in space. For instance, most of the glycolytic enzymes aggregate on the cytoskeleton of the skeletal myocyte. It's just like a block of protein that glucose comes in, pyruvate comes out. And this is also related to the notion of channeling. People talk about the enzymes being close to each other, and a metabolite is channeled through that in space. So you can map that on there. You can also map on expression profiling data from many, many different conditions for that organism and see the transcripts that represent the pathway are somehow coherently regulated. And you can go up to the gene level and you can look at the operons to see if there's a transcription factor or transcription factors that specifically regulate the functionalities of that pathway. What the panels at the bottom show there is mapping of available data sets like this in yeast and E. coli, and I believe also there was a human application in here too, to see if the data sets, these data sets that are independent of calculating the MinSpan are consistent with its structure. And they are. And what is also shown in there is a set of pathways gotten out of KEGG, biopsych, and the GO classification numbers. And in every case, the MinSpan represents the characteristics of these data sets much better than these other definitions, which are all man-made. MinSpan is computed based on the criteria that I've stated. Now, there are some notable differences between what the MinSpan calculated and what we might define by intuition. I stated earlier that for simple systems like glycolysis and pentose pathway, we can intuitively argue for meaningful pathway vectors. And we normally get something that is very close to what you would compute with a MinSpan. But when you get to scale, there are three features that pop up from the MinSpan that are different than the human defined pathways based on intuition or some biased view of the network that you're, may represent your personal interest, say. The MinSpan is objective, it's not subjective. So it discovers novel pathways, that is panel A. It also defines complete pathways. All the pathway usage and everything that comes with a pathway. It's not just methylation comes in and something leaves. That's not a complete pathway. So it's complete in that sense. It balances the map fully. And it also represents a nice way to modularize a network. And maybe I can jog your memory and see if you can remember a slide that I showed early in the class of the genotype-phenotype relationship being increasingly modularized into bigger and bigger modules. Here is a method to calculate objective modules that'll help you modularize that relationship. So it turns out that the MinSpan can be used to predict transcription factor activity. Because of this consistency that I spoke of, it can be used to predict transcription factor activity. We don't know all transcription factors and all the genes that they regulate, so we can now use this to predict and perform experiments. And this is an example of how that is done. So you pick a condition that is of interest to you, And this is done for glucose minimal media for the E. coli genome scale model. And you sample that solution space, and we'll talk about randomized sampling of states next time. And for every flux, you can get a distribution between two states. It's minimal media, and then that becomes your reference state. And then you may want to add, say, the availability of another nutrient and try to figure out what transcription factors will up and down regulate genes that are associated with that nutrient and having it available. So you can calculate these candidate distributions for all the fluxes under those conditions. You can map that onto the pathway matrix, the defined by the MinSpan. And you can calculate the weightings on each pathway. So you break down the most likely flux distribution into the MinSpan. You know, how do I have to combine the MinSpan to get that most likely state for the two conditions I'm comparing? Then you look at the MinSpan and see which pathways are changing the most, which alphas are changing the most, and then you can, based on that, look at the genes that have to go up and down, and look at all the promoters, and associate transcription factors with that and do a prediction. That's kind of what's shown in panel B. And the big complicated matrix there, the heat map, shows a large number of known transcription factors and their activity in E. coli, and the columns there represent different growth conditions. And you can do this comparison between pathways with and without the nutrient that would be shown as rows. No, those are the transcription factors shown as columns in here. And you can color code that based on what is known about the activity of that transcription factor. And you see a clear association between transcription factors and members of the MinSpan. But then if you look at the individual MinSpan that is associated with that transcription factor, and you will see that many of the genes in that MinSpan are not regulated by that transcription factor. And that sets you off on an experimental pursuit to see if that transcription factor or transcription factors regulating that MinSpan also regulate those genes. And such experiments were done, and a bunch of new such associations were found. So this is a very hopeful result. This particular set of basis vectors, because it seems to synchronize with all these other macromolecular data types we have information on, and seems to synchronize with the transcriptional regulatory network, at least as we know it in E. coli, and that these vectors are indeed the dimensions in the space that the cell is trying to regulate. And remember, it's sparsified, so these are the simplest vectors, and this would be the simplest structure of an operating system for that metabolic network. Okay, so that is a lot of detail about one particular and very promising set of basis vectors for the confined null space of a genome scale matrix, or actually more than one. We looked at E. coli and yeast in this case. Let me say a few more things about the set of basis vectors that I think is important. So we can calculate the MinSpan for the core E. coli network. The null space is 23-dimensional. There are 23 MinSpans in there. There is one type 3, so flux through that has to be 0. Turns out there are several pathways associated with the fermentation pathways. There are several of them associated with the pentose pathway and ETS functions. There are a set of pathways associated with the TCA cycle. And when you look at the structure of the MinSpan, and that's shown in panel A, you will see that there are 8 primary biochemical dimensions in it. It's not really kind of 23-dimensional. And that stack of pathways represented by the green branch in that classification tree are all glycolysis associated and fermentation product, fermentation byproduct secretion associated pathways. And then there are some associated with the TCA cycle and so forth. So the MinSpan, even after we calculate it, and it has mathematical dimensions that exactly correspond to the size of the null space, the dimensions of the null space, when you start looking at them, their biochemical dimensions effectively can be lower. And you can get at this by doing SVD, for instance, of the pathway matrix now, to see what, what subset of these pathways spans most of the null space, as an example. So there are some points to note here. Pathway vectors are a contiguous path through a network, and this is a characteristic of incidence matrices. When you start out, you can maybe mathematically define a bunch of such basis vectors, and they are non-unique. The steady-state flux vector will always be a linear combination of these basis vectors. And this is kind of the relationship between topology and function. Remember, we talk about a network in a one topology but many functions, and these basis vectors provide you that relationship. They tend to grow with network size. The new biochemical capabilities that are generated don't grow as fast as the dimension. And we saw that in the simple example I started out with when I put the purine pathways into that simple network of glycolysis and pentose pathway. There was really only one function of purine degradation, but 3 pathways, kind of similar ones. So there's maybe a dominant pathway that represents a function and a few around it that are kind of redundant but associated with it. And then, of course, the internal pathways, type 2 and 3, either they just have to be 0 by thermodynamic constraints, or 2 have to be downregulated just by physiological need. Okay, so let me spend the last few minutes then to put what we have spoken about here in context. And I like this quote from Uwe Sauer at the ETH that is shown there. He gave a talk here in 2005, and he had that comment on one of his slides. He says, pathways are concepts, but networks are reality. And what he meant by pathways there are the ones we see people draw in textbooks or other slides. They're concepts. But in reality, there's a network there, and the pathway has to be a part of the function of that network. And that is now, that divide is really bridged by these extreme pathways, and MinSpan is one example of them. So basically, biochemical functions tend to be discovered reaction by reaction. We know the historical examples of the pathway, the reactions in glycolysis, say, or amino acid biosynthetic pathway, amino acid biosynthetic pathways. And as more and more of them were discovered, you would string them together and define a pathway. And there are some famous cases, of course, historical associated with that. And maybe the most memorable one was the discovery of the TCA cycle, being a cycle. The predominant thinking before that was that these were all linear pathways. And you probably, forget his name now, heard about his dream of a snake that bit his tail and formed a cycle. And that's when he woke up, he realized, oh, this is a cycle. So that's kind of historical, but now, by having a genome scale view, and if we had perfect genome annotation, we can reconstruct and define the entire network. And pathways are now no longer concepts we can draw out based on our own preferences, but they actually become properties of the networks that we put together. And this slide here also reiterates what I stated earlier, namely that convex basis vectors, the extreme pathways, are not chosen by us. They're computed from the information that we now gather to put together network models. Let me end by making a few comments about this now direct correspondence between mathematical concepts and biological concepts. Okay, so first, let me show a cone here with 5 pathways. And let's talk about mathematics and biology. So the first thing I want to point out is that mathematically, this is a solution space for S dot V equals 0, and it's convex. And it's convex based on the constraints. So that's the mathematics. But on the biological side, the contents of that space represent the capabilities of that network, because any point inside that network represents a candidate stable homeostatic state. You can't be outside it, you can be within it. I think in evolutionary biology, this is actually called the reaction norm of an organism. which was kind of interesting. So the basis vectors mathematically for a convex set are unique, like I talked about. But on the biological side, we now see that these vectors, these convex basis vectors, are actually pathways, and we think of them as extreme pathways because they lie at the extremities of the space. Any point in that space is a particular solution, but on the biological side, it's a particular phenotype because that solution represents a functional state of the network, which would be an expressed phenotype. The flux vector is a non-negative linear combination of these pathways, so we can take any functional state and break it down into the pathways. And this was done, for instance, when we were trying to associate transcription factors to a pathway. You take the most likely flux state, break it down into its constituent extreme pathway vectors, and you carry out the analysis. So the basis vector has mathematical properties that we can study. The basis vector has biological properties we can study. Like we can compare what a mathematical dimension means to what a biochemical dimension means. So this is actually strikingly interesting in terms of mathematical biology or systems biology, that we have this one-to-one correspondence between biological concepts that we have known for a long time and the fact that we can now explicitly represent them mathematically. And those mathematics are specified by the genome scale reconstructions and the information we put into them. All right, so that is the end of the lecture. A few summary bullet points here at the end. So the stoichiometric matrix has a null space that contains all the steady state flux distributions. It is spanned by a set of basis vectors, which we can organize in a matrix called P. And if you multiply S from the right with P, we should get a 0, because every basis vector, every column in that matrix P is completely balanced. It's a completely balanced vector. We know now that these basis vectors correspond to biochemical pathways. We can form these basis vectors in different ways. If we view the space as being linear, infinite in all directions, we can always rotate the basis vector, the set of basis vectors any way we like. But once we put the constraints on the fluxes, that they have to be non-negative for elementary fluxes or for irreversible reactions, and they typically have a Vmax, we now close off the null space and form a confined null space. And that leads us to a set of basis vectors to span it that are convex. And we also got introduced to the fact that there are many of them. There are more of them than the dimension of the space. And with scale, when we go to genome scale, they become so many they may even be hard to compute. And there are many, many more than are meaningful in a practical sense. So we can define a subset of these extreme vectors, these extreme pathway vectors that are biochemically meaningful. And the MinSpan is probably the latest incarnation of such a definition. And it seems to be very consistent with macromolecular data types, as I showed you, and therefore represent a meaningful modularization of a network biologically. We also know, by looking at them, that the dimensions of this confined null space biochemically may be effectively lower than mathematically, because sometimes these basis vectors kind of bundle with similar biochemical functionality. So the dimension of the space can be maybe made hierarchical. There may be 8 major dimensions and then subdimensions within there. And if you want to now look at the pathway matrix, the matrix of these pathway vectors, you may now start to use something like SVD, like we talked about last time. I mean, how meaningful is the addition of every new basis vectors into that pathway? How much, additional functionality is represented by it? Some of that has been done, but not a lot. So that's the end. And the, I guess, take-home message here is that we now have mathematical objects or definitions that correspond with the biological ones that we're familiar with and we deal with qualitatively, but now we can deal with them quantitatively. And this is something we will build on throughout the class.